Good morning, everyone. This is the sixth lecture and the final lecture for your chapter Respiration in Plants. In the last class, we discussed the shuttle systems, which tell us how the NADH from cytosol is able to generate ATP inside the mitochondria. We also saw the net ATP gain from one molecule of glucose, as well as the percentage efficiency of aerobic respiration. In today's class, we will be discussing the final portion of your chapter, which is amphibolic pathway. Now, this is more of a compilation of whatever you have already studied somewhere. Okay, only a few things might be new, which we shall discuss. Otherwise, it is just your basic knowledge put together. Okay, let us see. So, here you have the three major respiratory substrates. In the first lecture itself, I had told you what respiratory substrates are, and there were two types of respiration. There was something known as floating respiration, and the other one was known as protoplasmic respiration. Floating respiration occurs when fats or carbohydrates are used for energy, and protoplasmic respiration takes place when the cell is in starvation condition and proteins are being used for energy. Now we shall start with the simplest one, which is carbohydrates, because we have just studied it in this chapter. So carbohydrates, whatever it may be, it could be glycogen, which is the storage form of polysaccharide in your animal system, or it could be starch, which is the storage polysaccharide in plants. Now, such sort of polysaccharides or disaccharides or oligosaccharides even, finally, what do they give you? After digestion, they give you simple sugars, example glucose. Now, this glucose or any other hexose sugar for that matter gets converted into glucose or is added to the same pathway, right? We have also discussed that point. So glucose here will undergo glycolysis, which is shown here, okay? Some steps are just shown with the arrows, okay? So glucose will enter the glycolysis by becoming glucose 6-phosphate. The enzyme here is hexokinase, fine. Then glucose 6-phosphate after a few reactions gets converted into fructose 1,6-bisphosphate. This is commitment step. Once it becomes fructose 1,6-bisphosphate, it cannot go back. Okay, it is destined to enter the glycolytic pathway. After fructose 1,6-bisphosphate, we have seen that it breaks down into two triose phosphate sugars, which are DHAP or dihydroxyacetone phosphate, which is a ketotriose sugar, and glyceraldehyde 3-phosphate, which is a aldotriose sugar. These two are isomers of each other and they are interconvertible. All the DHAP gets converted into G3P or glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate then after a few steps finally gives you pyruvic acid. This pyruvic acid by link reaction is converted into acetyl-CoA, and this acetyl-CoA enters the Krebs cycle. Krebs cycle finally gives you the carbon dioxide. Okay, even at the link reaction you have one carbon dioxide released, and this acetyl-CoA, which is basically two carbons. Okay, so two carbon dioxide molecules are released in Krebs cycle, as well as one molecule of water is released in electron transport chain. Clear? So this is your simple carbohydrate cycle, or rather pathway, right? Now, from this, let us understand the next part, which is fats. Okay, we will take the next respiratory substrate, which are fats. So fats are nothing but they are fatty acid and glycerol, right? So triacylglycerol or triglycerides or fats, they are all one and the same, right? TAG. If you remember, this is the structure. You have glycerol, which is a trihydroxy alcohol, right? So it basically has three OH groups. So 1, 2, and 3, and 3 carbons. So this is the glycerol molecule. These here are the fatty acids. Fatty acids are nothing but they are carboxylic acids, okay? And they have different chain lengths. So here, acid plus alcohol gives you ester. So this here COO is the ester bond, okay? And this way, one glycerol molecule can have 3 fatty acids attached to it. This whole thing together is known as triacylglycerol or triglycerides or just fats. So when these fats, they are digested, they give you fatty acids, which are these carboxylic acid chains, okay? So fatty acids plus the glycerol molecule. Now this glycerol, it is a 3-carbon compound, okay, and it has 3 OH groups. So after a few reactions, it can be converted into DHAP. If you remember, a part of this process we have seen in the glycerol phosphate shuttle. Okay, in glycerol phosphate shuttle, we have seen that the dihydroxyacetone phosphate can be converted into glycerol phosphate, and then glycerol phosphate can be converted into dihydroxyacetone phosphate. And the reactions are just dehydrogenation and hydrogenation. Okay, so same way, glycerol here can be converted into DHAP. So dihydroxyacetone phosphate. After becoming dihydroxyacetone phosphate, it can get converted into G3P and enter the glycolysis. Clear? So here can you see the connection? The glycerol part of the fats is connected directly to your glycolysis. At which point? At DHAP. This is important. Remember this point. Clear? Now let us talk about the fatty acids. This part I had covered when we were discussing mitochondria. Okay, mitochondrial matrix to be specific when we discussed cell. So the fatty acid, they undergo catabolism. Just like how this is carbohydrate catabolism. Catabolism is just breaking of something, right? So here the fatty acid catabolism is actually known as beta oxidation. And this beta oxidation takes place inside your mitochondria. What happens in the beta oxidation? Let me just show you very briefly. Okay. So in beta oxidation, inside your mitochondria, two carbons. Okay. This will be the first carbon. Let us say second carbon. So 2 carbons. Okay. They are removed one after the other. Clear? So in the first cycle of beta oxidation, you will have these 2 cycles being released. In the next cycle, okay, next cycle of beta oxidation, 2 more carbons will be removed. Then in the next cycle, 2 more carbons. Okay, so a pair of carbons, okay, so these 2 carbons, they are removed one after the other. So that is nothing but it is called beta oxidation, and it happens for fatty acids. Clear? Now what happens to these 2 carbons, okay, which are removed? These 2 carbons actually form what is known as acetyl-CoA. Clear? Acetyl-CoA, if you remember, is nothing but your acetate group or just 2 carbons which are attached to coenzyme A. Clear? So after beta oxidation of fatty acid, you get acetyl-CoA. And this is how fatty acid portion of the fats is connected to your glycolytic cycle. Clear? Are you able to understand? This is how they are connected. Fine, let me just erase that. Okay. So from fatty acid, by its catabolism or beta oxidation, you are getting acetyl-CoA. On the other hand, when there is enough energy, okay, what happens when you eat a lot of sugar, right? You get fat. How does this sugar get converted into fat? Sugar is carbohydrate and fat is lipid. So sugars, they get converted into acetyl-CoA, right? This pathway you know. Now this acetyl-CoA can actually be used for anabolic reactions as well. Anabolism is using small molecules and making a bigger molecule or a macromolecule, right? So one after the other, molecules of acetyl-CoA will keep on joining and form a new fatty acid, right? So this will be the anabolic reaction. From acetyl-CoA, you are getting fatty acids. Clear? So this is how fatty acids are connected to your acetyl-CoA. I hope till here it is clear. So this covers the fat portion of your respiratory substrate. Now let us talk about proteins. Proteins are heteropolymers of amino acids, right? So if you break down proteins, what do you get? You get amino acids. Now these amino acids, if they enter catabolic cycle, okay, if amino acids are further broken down, how do you break down amino acid? You remove the ammonium group Okay, NH3 plus is removed, COO minus group is removed. So if you keep on doing all of that, it can lead to different types of molecules. Some amino acids, after their amino group is removed, they get converted into pyruvic acid. Clear? So can you see here, amino acid catabolism is linked to the carbohydrate catabolism here at pyruvic acid site. Clear? Some of the amino acids get actually converted into acetyl-CoA. Fine? And some amino acids get converted into alpha ketoglutarate. Now this we have studied in the last class, if you remember the malate aspartate shuttle. Then glutamate. Okay, so glutamate is nothing but an amino acid, acidic amino acid, right? If from glutamate you remove ammonia group, okay, if you remove NH3 plus, then you end up with alpha ketoglutarate, which is also a 5 carbon compound. It is a 5 carbon organic acid, okay, which is a part of Krebs cycle, intermediate of Krebs cycle. So glutamate here, which is a type of amino acid, can get converted into alpha ketoglutarate, and whenever required, alpha ketoglutarate, if it is if ammonium group is added to alpha kg, it can give you glutamate again, right? So this is how amino acids, different types of amino acid, you have 20 types. So some of them get converted into pyruvate and can also be synthesized from pyruvate. So whenever, let us say, this specific amino acid is required, then from pyruvate itself you can convert it into amino acid. Example is aspartate. Okay, from pyruvate you can make aspartate. And I had shown you that how amino acids are interconvertible, right? Then from acetyl-CoA also you can make different sorts of amino acid whenever required. So that is why here double-headed arrows are shown, which means that the process can go forward as well as backward depending on the condition. Clear? So this is how all the pathways, they are interconnected. Okay, they are connected somewhere or the other. Now these connection points are very, very important to remember. Okay, the questions can be asked from here, they can be twisted, they can be in statement forms, or they can be here just like this given in the diagrammatic form. Clear? But you should be able to answer. This is just an example, not all of it is important for you to remember, but for you to grasp the diversity, okay, of this pathways. Let me just show you. This glucose, which we just see it as an energy molecule, can actually be playing a role in so many different pathways. Okay, let us see one by one. Glucose, we have already seen, it can undergo glycolysis for energy production. So glucose, it is getting converted into pyruvate, and then it can get converted into lactate in case of lactic acid fermentation. Clear? Now remember, during our cell lectures, I had discussed Golgi body, right? Or Golgi apparatus or Golgi complex, whatever. So this Golgi apparatus, it actually does the function of glycosylation. Glycosylation is what? Adding the carbohydrate moiety or carbohydrate molecule to your proteins as well as lipids. Clear? This is also taking place inside your endoplasmic reticulum at minor levels. So this glucose, okay, this glucose, it can get converted into glucose N-acetyl, okay, N-acetyl glucose, and then N-acetyl glucose can then be used to glycosylate the protein. This glucose, by glycolysis, it can give you acetyl-CoA, and this acetyl-CoA can be used for acetylation of the protein. So these are just protein modification, okay, from the normal structure of protein. When you add certain groups like phosphorus, if you add, okay, phosphate group, then it is called phosphorylation of protein. If you add this acetyl group in form of acetyl-CoA, then you call it acetylation. If you add some glucose-like molecules, then it is called glycosylation. Clear? So for all these processes also, you can see that glucose is very much important. Glucose is also important for nucleotide synthesis. We shall see it in the next slide how glucose can actually get converted into pentose sugar, which is ribose and ribulose. and ribose and ribulose can then give you different sorts of nucleotides. Glucose can also be used for phospholipid synthesis. We have seen phospholipids, okay, in biomolecules. So they are used there as well. Then glucose is used for fatty acid synthesis. We saw how glucose will undergo glycolysis. After glycolysis, what you get is acetyl-CoA. This acetyl-CoA will be used, okay, as a building block for making fatty acids. This is what I told you about getting fat. That if you eat a lot of sugar, you will say, oh, I'm not eating oil, I'm not eating anything else, I'm just eating sugar or I'm just eating carbohydrate. But still I'm getting fat. So this is the reason that all of the glucose you are eating, okay, whatever the excess glucose will be there, that excess glucose gets converted into acetyl-CoA, and acetyl-CoA is used to make fatty acid. What all these fatty acids do? They will form triglycerides, and triglycerides are storage lipids, okay? They are stored in your adipocytes, and that is why you can get fat. Then in cholesterol synthesis, remember this, cholesterol can also be synthesized from the acetyl-CoA. Okay, so acetyl-CoA, a lot of acetyl-CoA molecules, they will come together and they will form cholesterol. Clear? Triglyceride synthesis, I already told you, okay, from the G3P. Okay, G3P will give you DHAP, and from DHAP you can make glycerol, and with this glycerol, when you attach the fatty acids, you get triacylglycerides. Okay, then glycogen is the storage polysaccharide in case of animals. Then glutathione is a molecule which takes care of the oxidative stress. I'm mentioning this because it will be mentioned in the next slide as well. It is not much of importance in your syllabus, but it is important to know, okay, only for your general knowledge. So this glutathione, what it does is it takes care of the reactive oxygen species, which you call ROS. Reactive oxygen species is nothing but the oxygen which has a free electron and it can react and oxidize anything, right? So it takes care of such reactive oxygen species, ROS. So that is this glutathione molecule. Then, of course, we have seen that apart from glycolysis, the glucose can go into TCA cycle and give you more and more ATP, right? Complete oxidation. So from here only, you can say that glucose is such an important molecule. From the same molecule, you are getting so many different pathways, so many different biomolecules. So here are the alternative fates of glucose carbon. So what is happening to the carbon which is present in the glucose? Alternative, okay, apart from glycolysis TCA, what else can happen to the glucose carbon? Here just for your information, okay, not to mug up or anything, I have just shown you all the different amino acids, okay, like alanine, glycine, threonine, all of these can form pyruvate and vice versa. Pyruvate can form these amino acids as well. Okay, so at what point different amino acids can be made, that I have shown in this diagram. Right, so what is important for us is to find out the fates of glucose carbon. So intermediates of respiratory pathway branch off to synthesize, that is from whatever glycolysis or this TCA cycle, the intermediates, okay, these intermediate molecules, they can come out, they can branch off, and they will be used to synthesize amino acids as shown here, pentose sugars as we shall discuss, nucleotides. From the pentose sugars, of course, you can make nucleotides. Then porphyrin, I had told you, it is an organic compound, it is an organic molecule. And the structure we have seen in chlorophyll as well as hemoglobin. Okay, in hemoglobin, the heme group is porphyrin. In chlorophyll, also the main group, okay, the cyclic form that you see is porphyrin. Then in cytochromes, okay, cytochromes, remember, from your electron transport system in respiration as well as in photosynthesis. So these all cytochromes, they have porphyrin ring, okay, and we have also seen like cytochrome C will have C type of porphyrin or heme. Then cytochrome BC1 will have B type, C1 type. Cytochrome B6F will have B6 type and F type. So all different kinds of porphyrin ring. Okay, so these porphyrin rings are also produced from glucose. Then fatty acids, which we just saw previously, cholesterol, lignin precursor. Now, this is a very favorite question of some people. Okay, they keep on asking in some exams that, like, find out which of the following is not a polysaccharide or which of the following is not a carbohydrate. Such kind of questions can be asked. In which case, they normally mention something like pectin, hemicellulose, cellulose, and one of these options will be lignin, because lignin is actually a steroid type of molecule. Okay, and so it is basically like a fat. Okay, a different type of fat type of molecule. Fine. So it is also produced from your glucose. Then different precursors for carotenoid we have seen in accessory pigments, right? And different hormones also can be produced from glucose carbon. Clear? Fine. So now this is one small topic which I would like to cover, just in case if any question gets asked from a related topic, okay, or just to make you clear of this topic that we are studying today. This pathway is known as hexose monophosphate shunt, HMP, or it is also known as pentose phosphate pathway, triple P. Clear? Now, the details are not very much necessary. All you have to remember is this, you already know what is shown here in the cream color or whatever skin color. That is your glycolysis pathway. Clear? Which is very much clear to you. I know that. Now, what is different in this pathway that I want to show you? So here, um, as you might have already noticed that in photosynthesis, we were getting NADPH as reducing power, while in respiration, we have only seen NADH everywhere, right? So this is a sort of thumb rule to remember that during catabolic reactions, mostly you get NADH, and during anabolic reactions, you get NADPH. Okay, it is just a thumb rule. There are a lot of exceptions, but just to remember. So, when you want to synthesize, let us say, fatty acids, you require NADPH. So, in your body also, for a lot of anabolic reactions, NADPH is required. But we just saw that during cellular respiration, only NADH was synthesized, or FADH2. So, how is this NADPH produced? The answer is HMP shunt or your pentose phosphate pathway. Just focus here. The first step is the same, just like glycolysis. Glucose gets converted into glucose 6 phosphate. Now, this glucose 6 phosphate gets converted into another molecule, okay, which is known as phosphoglucolactone. The name is not important for you, just know that from glucose 6 phosphate, another molecule can be produced, and during this process, one molecule of NADPH is synthesized. Okay. Now this 6-phosphogluconolactone gets converted into ribose 5-phosphate. Okay. And how does this happen? So there is one carbon dioxide molecule which is removed, and the hydrogen which would be attached to this carbon is actually given to the NADP plus. So here you get one more molecule of NADPH. Clear? So in two reactions only, you are getting two molecules of NADPH, one carbon dioxide, and one molecule of ribose 5-phosphate. Clear? So this is how from your hexose sugar, pentose sugars are made. Fine? Now this ribose 5-phosphate, two of these molecules, okay, now the C5C5 means two molecules of 5 carbon sugars. These two, there are some trans ketolases and trans aldolases. These are just enzymes. What do? They shuffle the numbers. Okay, so 5 plus 5, so you have 10 carbons in total. After shuffling, this enzyme, it will transfer some of the carbons from here and there. And after shuffling, it will give you a C3 and a C7. So it will give you a, what you can say, a 7 carbon sugar and a 3 carbon or triose sugar. Clear? Now 3 plus 7 it will again depend what your body or what the system is requiring. If it requires C3 type of carbon, it will get used up. If it requires C7 type of sugar, it will get used up. If both of these are not required, then again it will be shuffled with help of enzyme. So 3 plus 7 again is 10. This 10 is again shuffled in form of 6 plus 4. 6 is nothing but your hexose sugar, and 4 is your tetrose sugar. Clear? So this is how from a molecule of glucose you can get different varieties of sugars. Okay, you can get pentose sugars, you can get triose sugars, tetrose sugars, 7 carbon sugars. Clear? So this is how from one type of sugar you can get different types of sugars. Clear till here? Now this reaction, okay, this hexose monophosphate shunt or triple P pathway, it takes place in your RBCs also, red blood cells, because they function with a lot of oxygen. Okay, they have hemoglobin and they are taking care of a lot of oxygen. Wherever there is oxygen, there is a chance that reactive oxygen species may develop. Okay, and if reactive oxygen species are present, they can oxidize your DNA, they can oxidize some important molecules in your body, just so that it does not take place or it does not happen. This NADPH in your RBCs is used to make glutathione. So this is where the glutathione is important. Clear? Now this is just the simplest version of HMP that you should just know. Okay, that from glycolysis, the first step, after the first step, glucose is converted into something else. You get one molecule of NADPH. From that something else, you are getting 5 carbon sugar or ribose phosphate, and 1 carbon dioxide molecule will be removed. So from 6 carbon to 5 carbon, and one more molecule of NADPH. Now these NADPH are used for many different anabolic functions. Right, so this again we have already discussed. Acetyl-CoA will be used for synthesis of fatty acid, for cholesterol. Gibberellic acid, which is a type of plant growth regulator. Clear? Then succinyl-CoA can be used for synthesis of chlorophyll, cytochromes, phytochromes. Pyruvate, oxaloacetate, alpha-ketoglutarate can be used for synthesis of amino acids. Oxaloacetate for synthesis of different alkaloids. Alkaloids we have seen in secondary metabolites. Okay, so this is the main point. Now what we are studying till now, because the respiratory pathway is involved in both anabolism as well as catabolism, it would be better to consider the whole respiratory pathway as an amphibolic pathway. Okay, you cannot say that it is just a catabolic pathway. A lot of different intermediates are used for anabolism as well. So the pathway that we studied first is known as an amphibolic pathway. Okay, the one with fats, carbohydrates, and proteins, which I discussed at length. That pathway is known as amphibolic pathway, rather than just calling it catabolic pathway. Okay, amphibolic meaning it is doing both. There can be anabolism when required and catabolism as and when required. Yeah, chalo. After covering that important topic, we are now at the last topic, which is respiratory quotient. Quotient, wherever you hear it, is mostly like a ratio, right? So respiratory ratio, the ratio of volume of carbon dioxide evolved. Wherever you hear the word evolved, it means released. Okay, so amount or volume of carbon dioxide released to the volume of oxygen consumed. Now in respiration, what are we doing? We are taking in oxygen when we are releasing carbon dioxide, right? So the volume of carbon dioxide released, which will be above. Okay, so volume of carbon dioxide released divided by volume of oxygen absorbed. This is known as respiratory quotient or respiratory ratio. It is denoted by RQ. It is measured with help of such type of a contraption, which is known as Genog's respirometer. Okay, and let us just see what kind of what you can say experiment you can do. Okay, to find out the RQ. So let us say you have taken this test tube. Fine. This test tube has this sort of a vial. Okay, vial is nothing but a chamber, let us say. This chamber has seeds which have been soaked. Okay, so soaked mung seeds or soaked any seeds. Fine. Now beneath these mesh will be nothing but a net-like structure. Okay, so in the net you have these seeds. Below the net you have KOH. KOH is potassium hydroxide. If you remember from your photosynthesis lectures, you must know that potassium hydroxide has this tendency of grabbing carbon dioxide. Okay, it attracts carbon dioxide. And if you want to know the reaction, potassium hydroxide, which is KOH, gets converted into K2CO3, okay, potassium carbonate. Fine. So this is the kind of structure you have, or the setup you have created. From this, you have to focus on this tube. Okay, there is this one single tube which is coming out. Forget about the syringe and all for now. There is this tube which is coming out. In this tube there is connected a U-shaped capillary. Okay, this U-shaped capillary is connected. Wherever you see this, it is a rubber tubing, okay, which is connecting these two tubes. In this capillary you have filled it with either colored oil or some other thick viscous liquid, okay, which is colored, so you can easily see it. Fine. On the other end it is open, okay, so it can easily displace, it can easily move. Fine. Now with this U-shaped capillary you can also see a scale. Okay, can you see the scale which is present there, right? So from the scale you can measure how much liquid is going up or how much liquid is coming down. Fine. Now what is happening in this setup? The setup should be clear to you by now. As these seeds, okay, they are soaked. So what will happen? They will respire. Seeds are also living things, okay? They are not non-living things. So when they respire, what happens? They take in oxygen, right? And there is no other place to take oxygen from because this tube is sealed. Where else can you take oxygen from? So you can take only from this capillary. So oxygen will be taken up from this capillary. Clear? So when these seeds, they take up oxygen, and of course because they are respiring they will release carbon dioxide. This carbon dioxide gets collected here, okay, because the potassium hydroxide, it takes up all the carbon dioxide. So what happens? As much as oxygen this seeds will keep on utilizing, this liquid here in the U-shaped capillary, okay, in your U tube, it will get raised, okay, the level will rise. Why? Just think of it like a straw. If you suck shake from the straw, the shake will rise, right? Same thing is happening here. The oxygen is being utilized. Because a lot of oxygen is being utilized, this level, okay, it will rise. What you have to do is at the beginning of the experiment you have to note down this measurement, and at the end of the experiment you have to note down the measurement, how much the liquid has risen. Okay, then using this simple formula of cylinder, okay, volume of cylinder, if you know how to find out, you just require the radius of this capillary. So the radius is normally printed on the capillary, okay, or you know the radius of this capillary. Then you know the H. H is what? It is the displacement, which is final minus initial. So that will give you how much this liquid has displaced. So you get displacement H. Then you know the value of pi. You know the value of radius, and you know the value of height. So you just have to use this formula pi r square h. Okay, and using this formula you can find out the volume of oxygen which is consumed. Okay, the amount of oxygen, the volume of oxygen which these seeds they took up. Okay, and by weighing out this solution here you can find out the amount of carbon dioxide that was released by the seeds. Once you find both these values, you just have to put them in this formula, okay, volume of CO2 by volume of O2, and that is how you find out RQ. In case of insects also you can do this, okay, in other live system also you can do. As you can see here, oxygen it will take up as much as oxygen it is taking. The levels, okay, it keeps on increasing. Fine. And here the carbon dioxide this insect is releasing is getting converted into K2CO3, okay, by reaction with KOH. Fine. Now, what is the importance of this RQ? Why do you want to find out the RQ? Because by finding out the type of RQ, you get to know what substrate is being used by the living system. Okay, so if you do this experiment on yourself, okay, there are certain respirometers. You just have to put a tube in your mouth and you have to breathe in, you have to breathe out. And there is mass spectrometer and other instrumentation which will just analyze. Okay, in the computer itself, it will show you the graph of carbon dioxide released and oxygen consumed. From this, they can come to know whether your body is burning fat, or is it burning protein, or is it burning carbohydrate. Clear? Chalo. So for carbohydrate, the RQ, respiratory quotient, is 1 because equal volumes or equal amounts of carbon dioxide are released and oxygen is consumed. Clear? Example is glucose oxidation. Remember that example. C6H12O6 plus 6 oxygen gives you 6 carbon dioxide and 6 water. So if you see here, 6 CO2 by 6 O2. So 6 and 6 get cancelled and you just have 1. Okay, so a respiratory quotient of any carbohydrate is close to 1. For fats, it is less than 1. Okay, so for example, palmitic acid, you must remember different sorts of fatty acids. Okay, palmitic acid is the 16 carbon long fatty acid. So C16H32O2. Okay, here when palmitic acid is oxidized, okay, when it is used for respiration, it will use 23 oxygen, give out 16 carbon dioxide and 16 water. When you see this 16 by 23, okay, 16 carbon dioxide released by 23 oxygen consumed, you find out that the ratio is closer to 0.7. Okay, it again varies with the type of lipid which is being consumed, right? The type of fat being consumed, fine, but it is normally closer to 0.7. So for fats, the respiratory quotient is around 0.7. So when the same experiment is done on you, and if the respiration quotient comes out to be 0.7, then the doctor or the scientist knows that your body is actually burning fat. Okay, it is important for dietitians and certain level of study to find out at what point of time your body is utilizing what uh, respiratory substrate. Clear? Then for proteins, the respiratory quotient is about 0.9. Here it is shown about albumin. Albumin you must have heard at many places. You have egg albumin, then there is albumin in your blood. Albumin is also present in the milk, which is known as lactalbumin, right? So albumin is just a globular protein and this is its formula. You do not have to remember for fats as well as for albumin. Just this is given as example, okay? That 63 carbon dioxide are released and 77 oxygen are consumed. So here, if you find out the ratio, it is closer to 0.8. Again, it can vary with the type of protein. Fine. So these are the 3 important respiratory substrate and their respiratory quotients. When is RQ less than 1? Okay, respiratory quotient is less than 1 when either there is complete oxidation of protein or fat. In case of fat, it is closer to 0.7. For protein, it is 0.8 to 0.9. During protoplasmic respiration, okay, in a starved cell which is undergoing protein oxidation, in protoplasmic respiration, the RQ will be less than 1. In case of a mixed diet, let us say your body is utilizing in mix, okay, some places it is using carbohydrate, some places it is using fat, in which case your RQ will show less than 1. And in case of germinating fatty acids, fatty seeds, my mistake. Fatty seeds are what? Like seed like groundnut. Okay, when you soak groundnut, and if you find out their respiratory quotient, it is less than 1. Why? Because they are fatty seeds. Okay, oily seeds, and they use fatty acid for respiration. Then there are places or types where there is respiratory quotient higher than 1. When is this possible? So for organic acids can be C4 type of acids like malate, oxaloacetate. Okay, here it is shown. Here is a malate. Malate plus 3 oxygen give you H2O plus 4 carbon dioxide. So when you find out the RQ or respiratory quotient, 4 by 3 is 1.33. Okay, so this 1.33 is greater than 1. So if the respiratory quotient is greater than 1, you know that the organism is living on organic acids. For alcohol fermentation, the respiratory quotient is infinite. Why? Because here no oxygen is consumed. Remember, it is anaerobic type of respiration, right? Fermentation. So from glucose, you are getting 2 molecules of ethanol and 2 molecules of carbon dioxide. So 2 molecules of carbon dioxide divided by 0 oxygen gives you infinity. Right. So again, the use is to find out the amount of oxygen consumed, to find out the metabolic rate, and to find out the type of food which is being metabolized. Now, factors affecting respiration. You can have light which affects respiration. How does light affect respiration? So actually, it affects photosynthesis, right? Now, if there is higher photosynthesis, then there will be higher substrate or higher glucose production, right? And if you have a lot of glucose, then indirectly your respiration will also be high. So if there is high photosynthesis, there is higher concentration of substrate or glucose, and hence there is indirectly high rate of respiration. After sundown, that is after there is sunset, okay, after sunset there is higher rate of respiration compared to right before sunrise. Why? Because after sunset, what happens? During the day, a lot of glucose might have been synthesized by photosynthesis. So all of this glucose is being used for respiration. Before sunrise, what has happened is all the glucose you have used. So the rate of respiration will become low. And after sunrise, of course, photosynthesis will take place. Then ATP, if there is higher concentration of ATP in your cell, then there will be negative feedback. Negative feedback is this ATP will go and tell that you do not require more and more respiration. Okay, it will go and tell your glycolysis that we do not require glycolysis anymore. We have enough ATP. In which case the respiration rate will become low. Clear? So high ATP concentration will lead to negative feedback or will decrease the rate of respiration. Carbon dioxide, if there is high carbon dioxide, it will reduce the respiration and decrease the plant growth, because carbon dioxide is actually the product of respiration, right? And by Lee Chatelier's principle, if you know that if on whatever side of the reaction, let us say if the products are high, then the reaction will go backwards or it will slow down, right? So here that is what is happening. Because the product, which is carbon dioxide, is higher, the respiration rate will be lower. Temperature, optimum temperature is 20 to 40 degrees, which is again very much optimum for your enzymes, right? So enzymes, they function better at 20 to 40 degrees. There is no respiration at 0 degrees Celsius. This is actually not true. There is very less amount of respiration. It is closer to 0. So you can just say there is no respiration at 0 degree or less, which is why when you store something in freezer or if something is stored under the ice, then it remains fresh for a longer time. There is Hoff's law, which again we have already studied in photosynthesis. It just says that there is 2 times increase in rate of respiration with increase in temperature by 10 degrees. So from 20, let us say you increase the temperature to 30 degrees, then the rate of respiration will become double. If you increase from, let us say, 30 to 40 degrees, then the rate of respiration will again become double. Fine. But of course, there is a specific range, and that range is 5 to 25 degrees. So between 5 degree to 25 degree, if you increase 10 degree temperature, then the rate of respiration will increase twice. And this is known as Hoff's law. Right. So oxygen, if the it is the concentration of oxygen which decides the type of respiration. If there is less oxygen, then of course it will go for anaerobic respiration. If oxygen is present, then the system will go for oxidative or your aerobic respiration. Cytochrome C oxidase, which is your complex number 4. Okay, this complex 4 is not sensitive to oxygen until 0.05% concentration is reached. So at least 0.05 percentage of oxygen, okay, should be present for cytochrome C oxidase to function. Then oxygen, because it diffuses very slow in water, okay, and your cytoplasm, your body is made up of mainly water. So inside your body it is diffusing very slow, okay, it is 10,000 times slower diffusing in water compared to air. So in this way also it can affect the respiration. So some plants, they have intracellular air system like parenchyma. Okay, parenchyma is a type of parenchyma. Parenchyma is nothing, it is a type of cells, okay, simple cells, soft tissue in plants. Now this parenchyma, between the cells there is a lot of space, and in this space oxygen will be, or air will be stored, okay. So this type of parenchyma is present in hydrophytes, which is the plants which live in water, or it is also present in roots of rice. If you know paddy fields or rice fields, they are normally flooded with water. So how do the roots respire, or how do the roots get enough oxygen? So they have parenchyma where they store oxygen or store air. Fine. Very low levels of oxygen lead to accelerated breakdown of sugar to ethanol, and carbon dioxide is evolved. Okay, so when there is less oxygen level, then more and more sugar will be broken down into ethanol or alcohol, and carbon dioxide will be released. While if there is high oxygen, then higher oxygen will inhibit the fermentation. Okay, so this inhibition is known as Pasteur effect. Pasteur is again that Louis Pasteur or Louis Pasteur rather. So this Pasteur effect is nothing but uh, wine industries. Okay, they actually called this Louis Pasteur, and they were like कि हमारी wine जो है it is becoming खट्टा, or it is not able to ferment properly. Okay, we are not getting enough ethanol. So what can be the reason? And Pasteur was the one who found out कि if you let the yeast, okay, break down glucose in presence of oxygen, अगर आप ये वाइन बनाने का प्रोसेस ऑक्सीजन की प्रेजेंस में करते हो, then it will lead to oxidative breakdown of glucose, and it will not give you ethanol. Okay, so oxygen, high level of oxygen, it actually inhibits fermentation. It does not allow fermentation to take place, and this inhibition is known as Pasteur effect. Clear? So remember this. इसलिए जो भी ethanol बनता है, alcohol बनता है, वो एकदम packed fermenters में या packed barrels में बनता है, if you have seen. Clear? Right. The final point in factors affecting plant respiration are the inhibitors. So just like photosynthesis, here also there are some inhibitors which can inhibit or block electron transport chain as well as oxidative phosphorylation. Okay. Complex wise, I have listed a few inhibitors. Here are the complex from 1 to 5, their names, okay, and the inhibitors. For complex 1, which is NADH dehydrogenase complex, rotenone and amytal are the inhibitors. These are also sometimes referred to as respiratory poisons. It is all one and the same. They are inhibitors, blockers, poisons. What they will do is they will block the electron transfer from complex 1 to ubiquinone. Fine. And for complex 2, there are no inhibitors which are required for your syllabus. Okay. For complex 3, you have antimycin. For complex 4, you have cyanide, azide, carbon monoxide. So if you know that, oh, if somebody eats a cyanide, then it is poison, that person will die. Why will that person die? Because the cyanide, it will actually go and block the transfer of electron from cytochrome C to oxygen. What will it do? It will take up that electron from the cytochrome C to itself. Okay, the cyanide will accept the electrons. It becomes the terminal electron acceptor. So that oxygen will not get converted into water. Okay, it will not accept the electron, and so the whole pathway, the whole electron transfer will get blocked. And if the electron transfer cannot take place, then Of course, ATP will not be generated, and if ATP is not generated, then your body starts to shut down. So cyanide, azide, carbon monoxide, these are all respiratory poisons. Okay, if you take this, or if by some mistake you are inhaling carbon monoxide, it is inhibiting your complex 4 or cytochrome C oxidase. Yeah, and finally you have FOF1 ATPase. Okay, this a lot of students call it F0 F1, which is also fine, but it is actually FOF1. Now, why this FO I'm saying? Because they found out that there is a inhibitor known as oligomycin. This oligomycin, it inhibits FO particle. Okay, so the particle or the part of the ATPase which is present inside the membrane. Clear? So that particle is inhibited by oligomycin. So it was given the name FO or F0, and later on the part which is coming out, okay, it was named as F1, the lollipop type of part. Okay, so F0 F1 or FOF1 ATP synthase. The oligomycin is the inhibitor which inhibits this complex. Yeah, and here we end the chapter. Okay, there are some things which are given in your material which I noticed, okay, which are just mentioned here if you want to find out. In some species such as spinach and pisum is your pea. Okay, so in pea plant and spinach, there is something known as cyanide resistance respiration, which means even in presence of respiration, even in presence of cyanide, the respiration will go on. Okay, they are not affected by cyanide, so that is called cyanide resistance respiration. And finally, there are uncouplers, as the name suggests, uncouple. Okay, to break the couple, to break the pair. So what kind of uncouplers are present? So these are the molecules which destroy the proton gradient. Okay, what they'll do? They will not allow the proton to gather around or get concentrated in the intermembrane space. So protons will be brought back from the intermembrane space into the matrix. And when that happens, your proton gradient is breaking down without producing ATP. So here you will not generate ATP. Okay, so these uncouplers are the molecules which will destroy the proton gradient, and thus the ATP production will be hindered. However, there are some such pesticides and some molecules known as dinitrophenol, okay, which do this uncoupling function. And there is a protein which does this uncoupling function known as thermogenin. Thermogenin is present in small babies, okay, because babies, they are very small in size, so they can get cold. Okay, so what happens is, in this process, if the proton gradient keeps on breaking, okay, without production of ATP, but there is a lot of heat generated during the process, this heat will keep the baby warm. Okay, so thermogenin is a natural uncoupler which is used by the body to keep the body warm. And yeah, we are finally done with the chapter, and I hope you had a good time. Okay, you understood all of it. Respiration is one of those topics where you can relate it to photosynthesis as well as biomolecules, which was why one of the reason why I wanted to cover it along with biomolecules. Okay, so biomolecules, photosynthesis, and respiration, so that all the molecules and all the pathways you can understand properly. If you have any doubts, please you can always ask me, and otherwise next time we will start with a new chapter.